First, I'd like to thank you and to thank the organization committee for giving me the chance to present some of my work. Actually, the main motive of these projects is the fact that metastasis is the major mortality cause of cancer patients. And in this brief talk, I will try to build the association or explain the association between Sialidosis X and A and metastasis, and our trials to study the regulation of Sialidosis X and A expression. So Sialidosis X and A are tetrasaccharides born on mucin proteins and also glycolipids of epithelial cancers, and actually they are the binding motif of some adhesion molecules called selectin family, and we are interested in the endothelial selectins E-selectin and G-selectin. And the difference between Sialidosis X and Sialidosis A is mainly the substitution on N-acetyl glucosamine on carbon 3 and carbon 4. So fucose and galactose are reversed on Sialidosis X, on Sialidosis A in comparison to Sialidosis X. What is the role of Sialidosis X in metastasis? Actually, in 2008, Saxton hypothesized the hematopoietic mimicry model, in which he stated that circulating tumor cells resembles immune cells in the way they extravasates the circulation towards inflammatory sites. So actually, circulating tumor cells expressing glycoproteins that has Sialidosis X will be able to bind endothelial selectins and then undergo subsequent adhesion interactions till extravasation and formation of the secondary mass of metastatic tumor. Not only that, it was proven, but also it has been proven that the first transition programs at the primary site also involve upregulation of Sialidosis X, and then these cells can be more invasive and more motile and undergo intravasation into the circulation. And also in the circulation itself, Sialidosis X helps circulating tumor cells to interact with platelets and leukocytes to form metastatic emboli. And actually this helps metastasis in two ways. The first way, it protects the circulating tumor cells to evade the immune system attacked by the immune cells, and also it can bridge the interaction with endothelial selectins, and all that will help the extravasation of circulating tumor cells to metastasize and form a new tumor at the distant site. And it's believed that Sialidosis X and its interaction with E-selectin is one of the factors that dictate organ-specific metastasis of epithelial cancer. But now we have a problem. Sialidosis X is a carbohydrate, and carbohydrates is not directly encoded by the genome, but they are biosynthesized with a specific group of enzymes called glycoside transferases. So in order to study the regulation of Sialidosis X expression, we need to study the regulation of glycoside transferases involved in the biosynthesis of Sialidosis X and Sialidosis A. They are mainly 4 enzymes, and we are interested in 3 major enzymes. The core 2 branching enzyme that puts the N-acetyl glucosamine branch on the protein, and 2 major terminal decorations, the fucose decoration and sialic acid decoration. So we are interested in fucosyl transferases, sialide transferases, and core 2 forming enzymes. And before that, we studied the expression of glycoside transferases in 2 breast cancer models, BT20, which is Sialidosis X positive model, and MCF7, which is Sialidosis X negative model. And actually we found a good disparity in the expression of glycosyl transferase between two models. As you can see here, the relative expression between BT20 and MCF7 shows some upregulation of some sialyl transferase, fucosyl transferase, and core2 forming enzyme. And now the question, how sialyl X is regulated, how the glycosyl transferases are regulated. So our aim was to to study the regulation at the transcription level, so to identify the transcription factors upstream of glycosyl transferase genes, and our hypothesis was that the transcription factors with higher frequency of occurrence upstream of glycosyl transferase genes will be most likely controlling this biosynthetic pathway. So for example, if I found that one of the transcription factors is controlling 8 isoforms out of 11, so most likely it's controlling the biosynthetic pathway. And in order to do that, we used the open source ENCODE ChIP-seq transcription factor data, and we used two inclusion criteria for the transcription factor ChIP-seq hits. The first is the transcription factor must lie within 1.5 kb window, 1 kb upstream of the transcription start site and half kb downstream of the transcription start site. And the second criterion is it must lie in DNase 1 hypersensitive region. And we started collecting the ChIP-seq hits and collecting the scores and we classified the glycosyl transferase genes into 3 classes. The first class is all the glycosyl transferases, or the 3 glycosyl transferases. So transcription factors in this group will be most likely controlling the whole pathway of the biosynthesis. And actually we got a repertoire of around 90 transcription factors. And the second group and the third group are the key decoration of the lactosamine unit, the fucose decoration and sialyl X decoration. And the top hits of the 3 groups actually including some, they include some interesting hits, like the major hits in the total glycosyl transferases are CMEC and ELF1 controlling 8 isoforms out of 11, followed by HEY1, FOXA1, FOXA2, MAX, and CBPB, which controls 7 isoforms out of 11. So maybe those transcription factors have a major role in controlling the biosynthesis of sialyl X. And in case of fucosyl transferases, after CMEC and DLF1, we found that again FOXA1 and FOXA2, CBP are controlling the expression of or lying upstream of fucosyl transferases genes, and in sialyl transferases we got TAF1 and YY1 and USF1. And actually we were interested in the question of what are cellular processes or cellular programs are involved in the or associated with those transcription factors and all our repertoire of identified transcription factors. So we did functional classification of the identified transcription factors, and we found interestingly that most of our transcription factors are involved in the morphogenesis programs and embryogenesis programs. And as you know that tumor cells at the primary site needs morphogenesis program at the first level to change from the stationary phenotype to the motile invasive phenotype. And actually this group includes the famous epithelial mesenchymal transition program involved in supporting metastasis of cancers. And also, interestingly, we found that transcription factors involved in the differentiation of immune cells, because actually immune cells share the same mechanism of sialyl X to extravasate when going to inflammatory sites. So actually this is very interesting. And actually we found some hits involved in proto-oncogene and in tumor tumor progression and metastasis, and interestingly also we found some hits involved in cell stemness. And actually this is outline of the transcription factor involved in three major cell processes: stemness, morphogenesis, and epithelium mesenchymal transition. We believe they are involved in controlling and supporting metastasis. So just to brief, Silyllus X forming glycosyl transferases lies downstream of a defined group of transcription factors like c-MEC, ELF1, FOXA1, FOXA2, HEY1, MAX. Those are the top hits of our study, and we believe that the identified transcription factors are associated with pro-metastatic programs like morphogenesis and epithelial mesenchymal transition, also associated with cell stemness, and actually there is a growing debate now in the community that cancer stem cells are one of the major factors of initiating distant metastasis. And also we found some hits associated with tumor progression and proto-oncogenes. This study, we hope it will permit further experimental investigations, and now actually I'm in the phase of preparing some stable transfectants of our top hits, and actually trying to do some expression experiments to correlate between glycosyl transferase expression and transcription factor expression. And our ultimate hope is that we can identify novel poor prognosis gene signatures associated with metastasis. Finally, I have to acknowledge the ENCODE project consortium and all my lab members and my advisor, Dr. Yasmin Mirzaban, and thank you.
Thank you for a very nice talk, a very detailed analysis. So as we said earlier, we will have all the speakers come up to the podium and answer questions that the audience has. So, Manolis, could you come up? Yeah, so if anybody has a question to begin with? Okay, so please come up here. So anyone has a question? Anybody has a question? Okay, please go ahead. There is a question upstairs among the ladies. Yes.
Do we have a microphone upstairs? Is there a microscope? Microphone.
As scientists, we always think about microscopes. Go ahead.
So I have a question for Adam. I'm wondering if you have tried to look at the influence of KD or KD prediction when you are assessing your protein networks. Is that something that is feasible given that you're already putting so much emphasis on simulation of these structures?
Yes, it is feasible. We thought about it, and one of the competing ideas to the thermostability of E. coli was the idea of pH influencing certain reaction pathways. But we haven't published anything, but it's totally feasible. Yes, we can, not only us, other people can predict to activities of specific groups and how a structure changes. So yeah, it's possible. It's something which could be done and should be done.
The reason why I ask this is because of a model system that I've been trying to work with is PDZ domains. So they're notoriously promiscuous, class 1, class 2, whatever. So I'm wondering where a single point mutation, not a silent one, but amino acid change, could affect significantly the KD. I mean, something that is like binding to something that is not. I'm wondering how much of that information are you losing by using these, domain similarity models? Do you get my question?
Yes, I understand the question.
It's kind of a part 2 of the same part 1.
So we can predict KDs. We cannot predict binding patterns. So with all these predictions, you have to be very careful with the questions you're asking. So for instance, we can answer questions about structural similarity. We cannot answer questions about docking specificity. They are very difficult to answer, especially high throughput. I can show you some slides where we tried and what level of errors we were getting. So at this point, at least in our hands, it's not feasible to predict docking specificity. So we can predict how KD of individual groups may change, but we cannot predict how it will change the interaction patterns. It would be great.
Yes, I am a clinical geneticist and I'm concerned about disease. We have here in our community, as we have a lot of consanguinity and a lot of disease, which is not as America or Canada or European country. So we have a lot, for example, we do a population a study in deafness, and we found the mutation which is causes non-syndromatic hearing deafness only 5% for connexin 26 and 30. So what did you think the evolution is affect our community also? We are in same time, not there is no evolution between us and Canada and the United States. So what's the answer of this different mutation we have?
This question is to whom?
To Dr. Kumar, Professor Kumar.
I learned the word evolution.
Yeah.
But it's difficult to understand. I could not understand your question very well. You said that
We have different mutation from Canada, from United States, from European. So in our country we have our own mutation. So I didn't think for myself, I didn't think evolution is lead to this different mutation. This is my thinking, because you said evolution is lead for a different mutation, and we have since long time a disease, and we think this is a mutation, and nowadays there is a different mutation. Did you think this be due to evolution? You didn't.
So let me answer one question on the population genetic side before Sudhir answers. So it is not true that Saudi population or other populations in general have different mutations. They do have many rare mutations that are unique. And that, of course, is not just Saudi. It could be any regional population or any isolated village or whatever. But the SNPs that Sudhir is looking at, these are SNPs that are generally shared and common. Down to 1% frequency. These are quite shared across populations and not infrequent. They vary in frequency, but their presence is not zero in most populations. So that's sort of the population genetics perspective, but clearly there's a lot of private mutations and very rare ones that will be unique to the populations. But my guess is that his framework could be applicable to those too, and he can answer that.
Yeah. Thank you for the question. So basically, always all mutations arise in a person, in one chromosome, right? And over time, some of them will spread in the population, that more people have that because of common ancestry. And then some of these mutations over time will be found in all the humans, and they become actual difference from humans to other species. So the process of selection, negative selection, or purifying selection, which sometimes we see as disease because nature is acting against the person with the mutation, is the process by which mutations are removed from the population if they're bad, and some of them are kept in the population if they have no effect. So the theory of molecular evolution applies from a person to a population, all the way to species. And so the project I mentioned was, it is easy to evaluate species to species differences because we can sequence all the species that we need close to humans, and using that information now, we can inform population and personal level, whether it is Saudi population, whether it is Indian population, whether it is American population. So the methods we developed apply to all variants, personal, family, and population variants, as long as the function of the gene remains the same and the position has the same function in the protein. So the application can be done in any population and for any personal or family or private mutation. I hope that explains the answer. You have another question?
No.
I think we will stop.
There was one more question. More questions?
I only want to comment, please, saying that mutation is different from SNPs. There are both variations, but they're totally different.
Well, mutation is what happens when DNA is replicated and error occurs. SNP is when you detect the mutation in a person or in a population, it becomes a polymorphism at certain level of frequency.
Yeah, but SNPs are the common things that happens, but mutation happens less than 1% of these variations, and they're rare. That's why we call them mutation.
Basically, the definition of what a SNP is, these days, anytime you observe a change in a person, you call it a SNP. So there is no requirement of 1% or 0.1% or 0.001% frequency. So essentially, when I say SNV, it is a variant, single nucleotide variant, not a polymorphism.
Yeah, that's right. And until you check that and you see how common is that in the population, and then you call it SNP.
Sure, that is the definition you can use, whatever. But this method that I developed and presented can be applied to any SNP or any mutation that is observed first time in the population. It has the same applicability, it's a general method. Okay, thank you.
Right, I would like to thank everyone. We will close this session. Hope to see you all sharp at 11:45, please, so we can start the next session. And as a reminder, the last session starting at 1:40 will be parallel. One will take place over here, and the other session will take place in the smaller seminar room. The posters are open for everyone to participate and have a look at. طيب جزاكم الله خير شكرا.
I guess this is a very selective audience. And in the next 25 to 30 minutes or so, I'd like to present to you some of our recent work on the genetics of rheumatoid arthritis. Now, all this data has not been published and has not been presented before. So if I did not make it clear, perhaps you will forgive me. Let me just begin with about rheumatoid arthritis, for those of you who are not initiated. Now, affect about 1% of the global population is a major disease, and the etiology is really unknown. Now, the two factors have been known for a long time to be associated with the RA. There's a rheumatoid factor, RF, and also ACPA, which is anti-citrulated protein antibody. Now, the genetic linkage is really weak. It is linked to HOA-DR4. It's with the identical twin study, it's about 12 to 50%. So, it is now deemed that the environmental factors, such as smoking, is associated with the severity of rheumatoid arthritis. Now, it's been known for some time that cigarette smoking has been closely associated with the severity of rheumatoid arthritis, and particularly these two seminal papers. So, the question that we pose is that what is the immunogenetics of cigarette smoking and RA? Now, let me just bring to you one
particular receptor called arrowhead carbon receptor. I think Professor Wu actually referred to that yesterday. Now, AHR is a cellular receptor of cigarette smoking. AHR activation promotes TH17 cell development. This is a very recent discovery. Two back-to-back papers in Nature about 4 years ago showing this one here. And then also, some years ago before that, TH17 cells has been shown to be a key pro-inflammatory cells in RA. So there is a complete linkage, the rational linkage there. Now, a word about AHR. It's also known as the deoxin receptor. It's a transcriptional factor of the basic helix loop helix per sim family. It's got quite a mouthful. AHR binds to its high affinity ligands, such as FICZ, translocate to the nucleus and dimerize by AHR, nuclear translocating factor AR and T, and could cause a variety of toxic effects. That's been known for a long time. The best studied downstream target of AHR is the CYP1A1 gene family that encodes the cytochrome P450 family drug metabolism enzyme. Again, all these are classical known AHR characteristics. Now, what is the association between AHR and RA? Now, we hypothesize that cigarette smoking aggravates RA by the activation of AHR. So, first of all, we'd like to look at the genetic polymorphism of AHR. And this work was led by a very talented postdoc fellow, Jimmy Tollett, and also Rafael Perez, and also his association with Professor Cunha in University of Sao Paulo in Brazil. So, the first thing we did is look at any association between the RA patient and AHR. Sure enough, we saw that healthy control and rheumatoid patient, when you do the quantitative PCR on CD4 positive cells, T cells, then you can see the rheumatoid patient here express significantly more the AHR compared to healthy control. Also, the downstream event of AHR, IL-17A, is similarly elevated, and also the TH17 cell, which is CD4 positive, IL-17 positive. This is not PCR, this is actually FACS analysis. And here again, the CYP1A1 in the quantitative PCR is elevated. So, RA patient express high level of AHR, IL-17A, and CYP1 in CD4 T cell than healthy control. That's the sense C. So then we look at the different SNP of the AHR. We'll cut this long story short. Just concentrate on this one to get this view here. RS2066853 has this lysin AGR and AG swap. And also as a control, we look at the intron SNP here, which is CT area. Now, looking at that, the important thing about the 2066853 is that it's been known for some time, since 2000, that AA and AG genotype in the SNP is increased AHR activity by increasing the binding of AHR to the downstream event in the AHR signaling cascade. So that's the reason why we have chosen this particular SNP for further study. Now you look at the SNP, so there seems to be some sort of a change here. If you have a TA and compared to the non-TA, you can see there is a significant increase in the disease score called DAX28, the disease activating score 28, looking at all the 28 joints in the body. And that is correlated very nicely with the increase of the ACPA antibody and the rheumatoid factor. Now, if we look at the, at the AGG patient here, And the patient with the AAAG phenotype has a significant higher disease score and also APA and rheumatoid factor. Now, then we look into the smoking people, smoking RA. This is a smoker and a non-smoker. And then you look at the smoking people patient has significantly more severe disease compared to non-smoker. And similarly in the ACPA antibody and also TS17 cell is significantly more as well. Now, hang on, thinking the same thing? Something. Okay, you look at the genotype then of the AA and AG phenotype, the genotype. Then you can see the smoker again has significantly more disease compared to non-smoker, and also in terms of ACPA and TS17 as well. So I missed something there. There is a very important slide which is missing. I don't know where it has gone to now. Now, the important slides that I have for you is this. If you stratify the patients according to the smoking and non-smoking, and also genotype AA or the non-AA, you find that the only patient who have exacerbated disease are the one with the AA genotype and smoking. If you have the AA phenotype and non-smoking, you are okay. If you have the GG phenotype rather than AA, then you can smoke as much as you like. It doesn't exacerbate disease. So another important message is that to get an exacerbated RA, you have to have an AA phenotype plus smoking. I don't know why this slide seems to appear disappear. All right, let's go on. So, what is the mechanism involved in such a combination? So we use the animal model and using what we call the antigen-induced arthritis model in mice, and also using, in this system, using methylated BSI, bovine serum albumin, as an antigen. Now this is a classical gold standard animal model of rheumatoid arthritis. Now, if we do this experiment, you either immunize them with PBS or methylated BSA using look at the naive animal. Now this is a mechanical threshold, is a measurement of pain. In other words, what you do, you put some weight on the pole. The lighter weight you need to make them withdraw, that means the more painful it is. So when you have a high, that means not painful. Low means very painful, in terms of the weight that you put on it. So if you immunize mice, you have pain showing. And then if you have the, what's this one here? Let's say sham. If you immunize the mice, you have the pain coming here. And if you sham, you compare the sham one. If you immunize the mice and put this FICZ, which is an agonist of the AHR, then you're more painful. Now, let me see, in the sham, in the immunized one, more painful. And then you could put FITZ in there, the immunized one is increased. In other words, the AHR agonist is reducing, is increasing pain. And that is also correlated with the neutrophil infiltration. I'm I'm sorry, the slides seem to be all screwed up. I can't show you the data here, but anyway, the neutrophil's profile is exactly opposite of the pain profile. Now, the AHR agonist exacerbates the AHR, and antagonist reduce it. Now, look at here, again, pain's profile here. If you immunize the mice, and then without this FITC, you get this sort of level. You put FITC in it. No, hang on a minute. This is ridiculous. You be here, and you put FITC in here, it gets more painful. But if you put the antagonist, then the pain is released. And you look at Th17 cells again, in the immunized mice, this is a completely seems to be scrambled. Right. We are actually using the AHR knockout mice here. So if you have the wild type mice, you have this much of Th17 cell. AHR knockout mice have less 17. If you put FITC into the wild type mice, you get that level of Th17. And in AHR knockout mice, you put the agonist, you do not get it. But if you now put CD4 T cell from a wild type animal into the AHR knockout mice, then you are rescuing more Th17 cells here. I'm not sure how I'm going to explain this one. Yeah. And right, let's try. I will say that AHR knockout mice develop less severe AIA, and AHR knockout mice fail to respond to FITC, but responsiveness can be restored by the wild type CD4 T cells, and this one seems to be okay. So then we neutralize Th17 knockout receptor knockout mice, and they fail to respond to AHR agonism. Here, let's see here. And just look at neutrophil here. This is a PBS control. You immunize mice, you get this level, and then you put FITC there, you get higher response in neutrophil infiltration. But if you take the antagonist of the AHR, then the neutrophil is reduced. And in terms of the Th17 cells, and this is naive mice, and this is immunized mice, the vehicle immunization here, if you put FITC, then you get increase, and take the antagonist, it's reduced. So I'd just like to repeat the conclusion. The 17 receptor alpha knockout mice fail to respond to AHR agonists in the AIA. So what we try to do is to simulate smoking in the mice. Now this is quite a tricky experiment. What you do, you put the mice into a smoking chamber and then introduce cigarette smoke. And the cigarette is Marlboro, which has open 8 milligram per cigarette of nicotine and 10 milligram per cigarette of tar. So if you do such experiment, look at the pain threshold. This is saline control, and if you immunize the mice, then they get more painful. And then you give them cigarette, 1 cigarette a day, 2 or 3 a day, you can see the pain increase in a dose-dependent manner. Now, I'd like to explain a little bit, is that we immunize the mice on day 0, and from day 12 to 17, when the disease already developed, you give them smoking, you put in chamber. And then you give them every day for 5 days, you stop. So this guy will have 1 cigarette a day, is 2 a day, and 3 a day. We can discuss the detail if you like. And if you look at neutrophil immunization, the infiltration in the joint, you can see that, again, the neutrophil infiltration is increased in a dose-dependent manner, dependent cigarette, the amount of cigarette you take. I'm sorry about the cigarette. This is a spelling mistake here. right? So, is there any relationship with 17? It looks like in this 17 case, the cigarette smoking increased the message of IL-17 in the lymph node of the mice, given the cigarette smoking. Now, this is another important message I'd like to go through slowly. So this is non-smoking and smoker mice. So if you were to immunize the mice, you get painful phenomena. If you put the antagonist of HR, then the pain is released. In the smoking one, again, the pain is higher than the control, and that, given the inhibitor, is reversed. And that is correlated nicely with neutrophil infiltration. And if you immunize the mice, the non-smoker, you have more neutrophil. You put inhibitor, that's reduced. And the smoker has more neutrophil infiltration, as expected, and they also can be reduced by the presence of the inhibitor. Then the messenger of IL-17 can also be used the same way. The message is increased by the number of cigarettes you smoke given to the mice. So the conclusion from this serious experiment is this. Cigarette smoking aggravates rheumatoid arthritis via the activation of the AHR. The genetic polymorphism of AHR, especially SNP 206-685, predispose RA patient susceptibility to cigarette smoking induced disease aggravation. The ligand of this cigarette smoke, which I'm going to present to you, includes halogenated aromatic hydrocarbons and non-halogenated polycyclic aromatic hydrocarbons. So, schematically, smoking activates AHR. The AHR activates TS17. TS17 induces inflammation, which contributes to the aggravation of RA. I must stress that cigarette does not initiate rheumatoid arthritis. The etiology of it is still unknown. What I present to you is that cigarette smoking can aggravate rheumatoid arthritis by the activation of AHR. And this AHR has to be a certain phenotype, a genotype, in such a way that they will lead to the increased activation. So the question that we're addressing now is it applicable to other diseases strongly associated with smoking, such as atherosclerosis? There has been a huge publication set associated with, by the epidemiology study, the atherosclerosis is linked to smoking, diabetes, as we all know, and that inflammatory bowel disease, IBD, and respiratory diseases, and periodontal disease, and also lung cancer. So we are now collaborating with various hospital and clinical group to explore these particular areas. So if you have any interest in this area, I'd be very happy to hear from you, and perhaps we can strike up some collaboration here. So lastly, I'd just like to acknowledge my collaborator. I mentioned Jimmy Tobin, Rafael Perez, and I haven't mentioned Jose Carlos Alves-Fioro, who is a key member in this area, and Paulo Rosata, who is the clinician, and Fernando Cunha, who is the head of the department here, and Peter St. Paulo. I have to mention that all these patients were collected in Brazil, in São Paulo region. Of course, there might be the population variation in other places as well. And the funding from the Brazilian source and also from the UK, World Trust, and MRC. So I'm going to stop here. Maybe I can take a question now or later this afternoon. Thank you very much for your attention.
Thank you. Because we have time, I take a quick question. Okay? Is there comments or questions? Okay, then let's move on, and we can take a question at the last moment if there is. Okay, the next speaker is Professor Ali Mobashir. His talk is about biomarkers of osteoarthritis, translating information from in vitro cellular systems to human tissues.
Thank you, Mr. Chairman, distinguished faculty, ladies and gentlemen. Thank you to the organizers for inviting me to come and present at this prestigious conference. I'm jointly appointed at the moment between the University of Nottingham and the University of Bradford, and I'm also part of the visiting faculty here at King Abdulaziz University. In addition, I work with 3 other centres that I'd like to mention here, because the context is relevant to my presentation. These centres are funded by Arthritis Research UK, and the core focus of these is arthritis. Each centre focuses on different aspects of arthritis. One focuses on pain. We have a centre that focuses on sport, exercise, and the risk factors for osteoarthritis. And the one at the bottom is a joint centre between Nottingham and Birmingham, and this one is exclusively focused on the concept of musculoskeletal aging. In addition, I'm coordinator for D-Board, but I'll tell you about that in a moment. So, what I want to talk to you about today, I'm not going to show a lot of data. I'd like to give an overview of the area of osteoarthritis, biomarkers, and why we have dedicated so much of our time and effort in this area. Osteoarthritis is a major emerging health problem. It's one of the most common forms of arthritis, more common than rheumatoid arthritis, and a major cause of pain, inflammation, and loss of mobility in synovial joints. It's characterized by loss of articular cartilage and inflammation in the joint, and as you can see from the Yeah, thank you. As you can see here, this is a relatively normal femoral head, and on the right-hand side we have an osteoarthritic femoral head with most of the cartilage worn away and the subchondral bone exposed. Now, the disease primarily affects load-bearing joints such as the hips and the knees, but other joints such as the hand, elbows, and shoulder can be affected as well. As I said earlier, it's a very common disease affecting approximately 10% of the population. No cure is currently available, and it's been designated as a priority disease area by the World Health Organization and various other funding agencies across the world. It's a major cause of disability. It's one of the top 5 healthcare issues in Europe and in the US, and is rising and is going to have a major economic impact across the world. So I'd just like to summarize some of the risk factors. Of course, age is one of the key risk factors, and beyond the age of 60, many of us will have osteoarthritis in one or more of our joints. But obesity and having a BMI of greater than 30 is certainly one of the contributing factors. Damage, injury or trauma to the joint, excessive loads on a normal joint can be a major contributor, or joint instability, normal loads on a joint that has previously sustained an injury. Many of the speakers in this conference have been talking about genetics. There are a number of genes that are identified using GWAS approaches. A number of genes are potentially linked to the development of osteoarthritis. but there are potentially metabolic and endocrine contributing factors as well, and there's a link with diabetes also. Now, joint trauma is one of the key contributors to the development of osteoarthritis, and participation in sports that involve contact, collision, and excessive force on the joints are key risk factors. Sports such as rugby, football, even tennis, professional tennis, running are all potentially going to be high-risk sports. And what I've highlighted here is some of the mechanical factors that can contribute to osteoarthritis development in athletes. Injury itself, surgery which occurs after the injury, the muscle weakness after the recovery period, and the subsequent joint deformity, and what might happen in the joint which becomes unstable with repetitive joint loading. So obesity is also a major contributing factor, and this slide really highlights the sort of League of Nations when it comes to obesity. And it's a very interesting perspective looking at the correlation between the prevalence of obesity and the prevalence of osteoarthritis. Clearly, in countries such as the United States and the UK, there is a significant correlation between osteoarthritis and obesity. But it's not just excess weight. A lot of investigators in this area are beginning to realize that mechanical overload is one of the contributing factors, but it's the systemic inflammatory mediators and the pro-inflammatory cytokines produced by white adipose tissue that are making a major contribution to disease progression. So if you look at the main tissue that's affected in osteoarthritis, articular cartilage, this is what it looks like in the normal state. Smooth surface, healthy cellularity, and a single interface between the cartilage and the subchondral bone. And here on the right, you see disease. Fibrillated, fragmented articular cartilage, evidence of cells that are undergoing cloning and reparative processes, duplication of the tide mark, and significant vascular and neural changes in the subchondral bone. Here, in this situation, we have homeostasis between extracellular matrix synthesis and degradation, and in osteoarthritis, we have predominantly extracellular matrix degradation and cartilage collagen fragmentation. Also, the behavior of the cells changes. They become significantly aggravated and inflammatory in the osteoarthritic joint. When we look at the balance between cytokines and growth factors, the so-called catabolic and anabolic factors, there is a significant shift towards the ones that induce matrix degradation. And the ones that are involved in anabolic reactions are reduced in terms of abundance and activity. So that brings me to biomarkers. Why biomarkers? What are biomarkers, and why do we need them for osteoarthritis? Let me just give you a very brief definition of biomarker, which is a characteristic that's measured either as an indication of a normal physiological biological process or a pathogenic response to disease processes or responses to drugs. And this definition has been around since 2001 and hasn't really changed significantly. There have been various refinements. Now, when it comes to osteoarthritis diagnosis, radiography is the gold standard. However, this gold standard is not adequate. By the time osteoarthritis is detected, there is significant joint damage and extracellular matrix changes in the cartilage. And the joint has gone potentially beyond the point of no repair. So there are significant anti-inflammatory outcomes in a joint that exhibits changed joint space width. Here you see a normal joint on the left and an osteoarthritic joint on the right. and the change in the distance between the bones above and below is actually one of the key measures that's used in radiography. But this is an indirect measure of alterations in articular cartilage. It doesn't measure a dynamic process. It takes a snapshot at a particular moment in time. Any changes over time are subtle, small, and may occur in a small subset of patients, the so-called progressors. Radiographs can be poorly reproducible, and most importantly, the correlation with pain can be questionable. So in certain patients, the joints are extremely painful, and in others where there's significant cartilage loss, there is no pain. So when you think about biomarkers, they are very useful tools to assess the existence or the progression of different diseases. And I've borrowed this slide from one of my collaborators at Nordic Bioscience in Denmark to highlight some of the targets that they've developed and that they're working on in different disease areas. And the ones that are particularly interesting for us are the markers of the rheumatological markers and the markers associated with changes in muscle. Now in osteoarthritis, biomarkers have a capacity to be able to detect changes early, before they've appeared on the radiograph. They can give us information about how the disease is progressing and predict the outcome of the disease. And when it comes to the different stages of osteoarthritis, it's not quite as complicated as rheumatoid arthritis. There are three distinct phases: a molecular phase, a pre-radiographic phase, and a clear recalcitrant radiographic phase. Now, in the molecular phase and the pre-radiographic phases, there is no pain. So the patient doesn't know that there is a disease process within the joint. And of course, by the time we get to the third stage, there have been significant and extensive changes in the articulating structures. And that presents us with a difficulty, because by that point, repair is going to be either difficult or not achievable. So we need a better gold standard. We need molecules, and we need diagnostic kits and assays that can give us early warning of joint degeneration and be able to give patients and their physicians an earlier and more targeted way to approach disease management. Also, biomarkers can facilitate drug discovery.